Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, we are discussing about the tools of recombinant DNA technology. Various enzymes are useful in recombinant DNA technology for various purposes. Today we shall discuss about two different enzymes. One is alkaline phosphatase and the other enzyme is terminal transferase. First of all, we shall discuss about alkaline phosphatase. Alkaline phosphatase is also known as basic phosphatase. This enzyme is optimally active at alkaline pH. Alkaline phosphatase was discovered in 1923 by Robert Robinson. This is a homodimeric metal enzyme. Its molecular weight is 86 kilodalton. Each monomer contains 5 cysteine residues, 2 zinc atom and 1 magnesium atom, which are crucial to its activity. Enzyme alkaline phosphatase is present both in prokaryotes and eukaryotes. Its basic function is dephosphorylation. Alkaline phosphatase removes the terminal phosphate group that is present at the 5' end of nucleic acid. So, by removing the terminal phosphate group from the 5' end, alkaline phosphatase prevent recircularization of linear vectors during cloning. Friends, this is the diagram of a plasmid vector which has been cut open by using a restriction endonuclease. So, you see here, this is the 3 terminus and this is the 5 terminus. At 3 terminus, this is the hydroxyl group, and at 5 terminus, this is the phosphate group. So, phosphodiester bond formation occur between 5' phosphate group and 3' hydroxyl group. If we will remove the phosphate group, then the formation of phosphodiester bond is not possible. So, by removing the terminal phosphate group from the 5' end, alkaline phosphatase prevent the recircularization of linear vectors during cloning. By removing the terminal phosphate group, alkaline phosphatase also allow for radiolabeling. This enzyme can also be used as a label for enzyme immunoassay. Friends, this is about alkaline phosphatase. Now we shall discuss about terminal transferase. Terminal transferase is an enzyme that is also known as terminal deoxynucleotide transferase (TDT). This enzyme was discovered independently by Bollon and his colleagues and also by Krakow et al. Terminal transferase is a specialized DNA polymerase. It is template independent. Its function is to add nucleotides to the 3' terminus of a DNA molecule. The preferred substrate for this enzyme is 3' overhang. 3' overhang is the preferred substrate for terminal transferase to add nucleotides. This enzyme also adds nucleotides to the blunt end and to the recessed end. This enzyme is expressed in immature pre-B and pre-T lymphoid cells. This enzyme is also expressed in acute lymphoblastic leukemia and lymphoma cells. Terminal transferase is the least understood enzyme of all polymerases. This enzyme can catalyze the synthesis of 2 to 15 nucleotide polymers from free nucleotides in solution in vivo. Like other polymerases, terminal transferase also need a cofactor. Cobalt ion is the necessary cofactor for this enzyme. but in vitro in solution. This enzyme can also work in the presence of divalent magnesium ion, manganese ion, and also zinc ion. Next is application. Terminal transferase has applications in molecular biology. This enzyme is useful in RAS. What is RAS? RAS stands for rapid amplification of cDNA ends. This enzyme can also be used to add nucleotides that are labeled with radioactive isotope. For example, in case of tunnel assay. This assay is useful for the demonstration of apoptosis. What is tunnel assay? Tunnel assay is terminal deoxynucleotide transferase DUTP nick and labeling assay. This is an assay that is useful for demonstration of apoptosis. Terminal transferase can also be used in immunofluorescence assay for the diagnosis of acute lymphoblastic leukemia. This enzyme is also used in recombinant DNA technology for the conversion of blunt end to cohesive end. Terminal transferase can be used for the labeling of the 3' end of DNA. And this enzyme is also necessary or useful for the de novo synthesis of oligonucleotides. Terminal transferase is also useful for homopolymorphism. Friends, this is about terminal transferase. Please share, like and subscribe my videos. The subscription is free of cost. Thank you very much. See you again.